Hi, my name is Jillian Petrosky, and I am a graduate student at the Boston University Bioinformatics PhD program. And today I'm going to be talking about MAPT subhaplotype associations with chronic traumatic encephalopathy endophenotypes. So for a little background on CTE, chronic traumatic encephalopathy, or CTE, is a neurodegenerative disease that is associated with repetitive head impacts, or RHIs. CTE is characterized by the accumulation of hyperphosphorylated tau lesions, much like other neurodegenerative diseases. Most individuals with CTE have reported playing organized contact sports, such as American football, or have served in the military. However, not all athletes and veterans with a history of RHIs develop CTE, suggesting that there are other underlying risk factors for acquiring the disease. Therefore, investigating the possibility of a genetic predisposition to CTE may increase our understanding of disease development. Microtubule-associated protein tau, or MAPT, is a complicated and disordered protein found in the 17q21 region. It has been implicated in tauopathies such as Alzheimer's disease, but not yet CTE. The 17q21 region, including MAPT, has 9 main structural subhaplotypes due to a megabase long inversion resulting in H1 or H2 haplotypes, and several copy number variations, alpha, beta, and gamma, resulting in several subhaplotypes throughout surrounding genes. Due to MAPT's role in tauopathy and its propensity for mutation, we have investigated associations between MAPT subhaplotypes with CTE neuropathology and related endophenotypes, such as tau burden in different brain regions. 458 donors of European ancestry from the Understanding Neurologic Injury and Traumatic Encephalopathy, or UNITE Brain Bank, with known RHIs were evaluated for CTE status, stage, and tau burden throughout the brain, as well as evaluated for a prior history of dementia. Donors were genotyped on 5,000 SNPs across the 17q21 region, and then tools such as impute2 and shape-it were used to impute and phase the subhaplotype markers throughout the MAPT region. And then the subhaplotypes were ultimately determined with another round of shape-it. The subhaplotypes can be seen here in this figure that was largely based off of work from Boecker et al. In Nature in 2012. And then depending on the determined haplotypes for each individual, we then did association testing between CTE and related endophenotypes and their haplotypes. The data we used from this project, again, came from the brain donors that part of the Unite Brain Bank at Boston University, which is the largest tissue repository in the world focused on traumatic brain injury and CTE. All of the donors had a history of exposure, and 82% of them were former football players. We limited our analyses to individuals of European ancestry and had individuals that were diagnosed with each different stage of CTE. We also have a representative sample of donors with 0, 1, and 2 copies of the H1 beta 1 gamma 1 MAPT subhaplotype that we were most interested in based off our results from the methods previously described, which allowed us sufficient statistical power for association testing. And here are our results from this work. So our association testing of the H1 beta 1 gamma 1 MAPT subhaplotype with CTE and endophenotypes. To do this, we used a linear model with the dependent variable being the phenotype of interest and predictor variables of copies of H1 beta 1 gamma 1 alleles, age at death, and 10 principal components to control for population structure. We correlated our p-values for multiple hypothesis testing using a permutation test on account of our highly correlated phenotype outcomes. We found that the H1 beta 1 gamma 1 MAPT subhaplotype is significantly associated with dementia diagnosis and a semi-quantitative tau burden in 5 different regions of the brain: the amygdala, entorhinal cortex, inferior parietal lobe, dorsolateral frontal cortex, and the strongest association in the superior temporal cortex, where the H1 beta 1 gamma 1 allele is associated with 60% higher odds of severe tau buildup. And we would like to thank all of our supporters in this work, and thank you so much for listening.